Okay, welcome everybody. Good morning. And today you are the very lucky people, because we're going to talk about a biophysical technique that's universal, something that could be used for everything. So in the LMB, we're very blessed again to have two types of calorimeter where we're going to be able to measure the heat of processes. One is called it's going to be called the isothermal titration calorimeter. It's isothermal, means it's going to be the same temperature, and we're going to then measure reactions. And the second system is called the DSC, and that's a differential scanning calorimeter. And as you can actually guess, it's going to vary the temperature, and we're going to look at melting there and stability. So the reason why calorimeters are so good at lots of different, looking at lots of different systems, is because heat's a kind of a universal change that occurs in biological processes. So you can imagine there on the top, you've got folding, then you've got binding as well. And associated with these processes, there is equilibria here and here. So how do we then measure this? How do we relate this heat to the binding constants? And we need to really then understand the thermodynamic principles behind. So in the next kind of couple of minutes, I'm just going to go back to where you kind of probably learned all this, but at least then you actually find out that stuff you actually learned at school actually has some relevance here. Okay, so let's imagine there's an equilibrium here. Well, that's related to then the free energy change, and that's just a logarithmic then expression of that. So, and it's multiplied by the gas constant, multiplied by the temperature in Kelvin, and then it's a natural log. So what does that mean? Well, that means that when delta G is negative, it means that the equilibrium is above 1. In this case, above there, you can see that that means there's going to be more products formed. So this reaction is going to be favored. And what does that mean in energy terms then? We all talk about binding constants and it's two free, it's twofold, threefold. What does that really mean in energy? Well, if you get an order of magnitude change in equilibrium, it means that delta G is going to then rise by 1.36 kilocalories per mole at 298. But it gets a bit more complicated because the free energy is then related to the changes in enthalpy and entropy. And either of these reaction then could then be either driven or opposed by the relative changes in these two forms here, the enthalpy or the entropy. So what are these kind of terms, and what do they actually mean? Well, in an exothermic reaction, you can imagine that energy is given off as the system goes down into a lower state here, and then you would say that the enthalpy, delta H, is negative. So bonds are being formed, heat's been given off, and the system's then in a lower energy state. But of course, energy doesn't like to be just stuck in one way. And energy likes to be distributed in different ways without kind of a change in temperature. And that gives rise to disorder or entropy. And so entropy is essentially just a measure of the number of different ways the system can be arranged. And if you get an increase in the entropy, then that's kind of universal. And if you think about entropy, just go back this evening, look at your bedroom floor and see, is there entropy there? Do you have everything arranged on the floor drobe? And so the effect of entropy then is increased with temperature, and that's just by thermal motions. So we can see that any kind of reaction is then essentially a balance between the changes in entropy and enthalpy. And so here's an example here on the left. This is two drugs that are binding onto DNA. One's an intercalator, one's binding in groove. And if you look at the bottom there, then the graph here is the y-axis is delta H, and then the x-axis is minus delta T. And you can see on the intercalators, they've all got positive delta T minus T delta S, so that makes it unfavorable. But they've also got they've got a very negative delta H. So the delta H in that case is driving that reaction. The groove binders, well, actually then the delta H is positive. So that's actually disfavoring this interaction. But actually the change in the entropy is negative, and so that's what's driving the reaction there. But of course, what about the water? You're thinking, right, we always discuss these kind of dry molecules that are kind of coming together. So if you kind of look at the top there, when you've got an interface, proteins are then surrounded by water, or other molecules again. And then when you form that interface, then you're excluding that water out. So the water is then got a large change in entropy because it is free to kind of move. So what happens is that these kind of large subation changes that happen in biological processes lead to a very large energy needed to actually increase the temperature. And this is called a large heat capacity. And what that means is then that both the delta H and the delta S are strongly affected by temperature. And again, I'm going to give you a trigger warning as well, which is there's equations coming up. But don't worry about it at all. Essentially, all you have to really remember is that the delta H then is really the integral of the delta Cp, while then the delta S is an integral of the delta V over T. So therefore, they end up with then different dependencies. One is linear dependence, and the other is a natural law of dependence. So that when we actually look graphically what the changes are, in this case, you can see with temperature, delta H is going from positive down to negative with increasing temperature. And then that's being opposed then by the minus T delta S in red, and that's getting from negative up to positive here. So then you're getting a temperature dependence on the equilibrium here. Okay, so let's first of all just go into our first system, and let's just talk about the ITC system. So how does that actually work? Okay, so you've got, on the top there, you've got two chambers. One's got water or buffer, and then the one on your left then has an injector in there, which also stirs. And that's going into a cell containing your protein or other macromolecule. And so what's going to happen during this experiment is then you're going to start to inject a sample into the cell, and that's going to spin, reacting into small volumes. And what the calorimeter does, it tries to keep a constant temperature difference between the reference cell and the reaction cell. And it's got these feedback heaters here just to try and keep either putting more heat in or less heat in, try and keep this change in temperature the same. And then this power that it puts in is calibrated very well, so it can tell how much excess heat is taken up by the reaction or given off. So an experiment might look like this. You've got a lot of material in the cell, and you're going to inject, first of all, very, very small amounts, like 2 microliters, 0.5, 2 microliters into the cell. And so you're getting up a lot of heat. And then obviously as the material in the cell becomes saturated, you're getting less and less and less and less and less heat. Okay, and so then it means there's much less power that needs to be changed. In this case here, we can see that this is an exothermic reaction, and these dips here are a decrease in the power having to go in. And so what kind of heats are we talking about? Incredibly small heats. You can imagine that it's in the order of microcals. And of course, microcals is not an SI unit. So 1 cal is 4.184 joules. And essentially lots of the literature you'll see will be always in calories. And think about microcals, compare that then. So you put in your box of cereal where you can kind of see what the calories are on that. And typically, a microcal in these kind of experiments, it's raising 1 mil of water by 1 by 10 to the minus 4 degrees. So it's tiny. So all the experiments we're going to be talking about is called microcalorimetry, and not like, say, large-scale calorimeter where you put a gerbil into an icebox. Right. Okay, so the good things about it is it's a direct measure of the system. So you're getting that heat out of the process. You can then interpret the thermodynamics, and basically anything that will go together, hopefully you can measure heat. And I'll show you an example of a very, very unusual experiment we did, Chris did recently. You can also use crude samples of high turbidity. It's also label-free. There's no labeling, there's very, very little assay development. And it's also a non-destructive technique. So you can take the sample out after you've finished, dialyze it, and you can run it again. I'm also showing examples where you can get kinetic parameters as well, in special cases. Right, what's the disadvantage? Well, heat's being generated by everything. So it's actually, you're just measuring the heat, but you don't necessarily know where it's coming from. So, if you get buffer mismatches or there's some, your DTT is going off, then you'd still measure that. It is very, very sample hungry. Of all the techniques that we got, it's probably the one that needs the most sample, but it does give you some of the best results. It can be quite low throughput. We do have an auto ITC where you can essentially just rack up all your samples and just let it run. So that kind of gets over the problem of manual handling. Because then of the complicated in terms of where the heat's coming from, you need to be very, very careful in the controls and the data analysis, which we'll come to. And of course, you're using quite high concentrations of sample. Typically, you're working way above the KD in both the material in the cell and also what's been injected in. And of course, some proteins and macromolecules don't really like to be very, very highly concentrated. Some proteins don't like being stirred. They're very, very happy. You've purified them, you've got them quite nice. You put them into the cell, and of course, during the reaction, then there's a spinner spinning around, and then you come to the end and go, why is my trace very horrible? You take the sample out, you can kind of see it's just aggregated. It's very important because you're doing finer thermodynamics, you need to then have very, very high concentrations, and that's just not just doing Bradford, you need UV or AAA. So because you're injecting one sample into another, again, you need buffers to match completely, and that can be a problem if you've got obtained so in small molecules. You also need everything to be very, very clean. So you have to hope the person who was there before you didn't kind of just aggregate the protein in and just leave it. So it's very important then to just make sure you wash out the stuff. Okay, so here's a typical workflow for ITC. Okay, how much samples you need to be clean, you need to be accurate, equivalent buffers. Now, if you've got CTT in your system, try and replace it with TSEP. The thing will be is that with TTT, you should be able to, because it is got two cysteines there, or sorry, yeah, two cysteines, sorry, two SH groups, then you can actually watch it oxidize during the time. So we've got an ITC 200. So typically in the cell, you need about 200 microliters, but to actually load it properly, then you need about 350. Concentrations might be 10 to 50 micromolar. We'll see later what the opposite concentrations. And then in the syringe we're going to put in, you need a smaller volume, but it must need to be much higher concentration, as we'll see why recently, very soon. So okay, you've got a cell and you've got a syringe. Which one do you go? Which one inject into what? Do it both ways if you can. But the choice may be limited because you can't get one of your components up to the right concentration. Typical experiment might be start off at 20 degrees. It's always good to do initial water to water just to check the system, because if someone has left it all dirty or the system, and you get a horrible trace with water to water, then you need to clean it all out, rather than say you just put your protein in and at high concentration and end up going, oh, that looks horrible data. Oh, it's a bit dirty. Then typically in the injections, we do about 20 20 injections with 2 microliters. And the questions you have to ask yourself is, does it go to saturation? And what buffer controls do you actually need? So here's an example here. On the left-hand side, you can see a nice kind of curve. At the top there, you actually go to saturation. So when you're there on the right-hand side of that left-hand graph, you're getting very, very small heats. And what those represent is that it's not the material that's been injected in, that's not binding to anything in the cell. That's just the heat of dilution, going from a concentrated solution then into the weak solution. On the right here, then you kind of get to the end, you kind of go, well, I've still got quite substantial heats. Does that actually need, do I need to have different concentrations to go on? Or is there some kind of mismatch? So then what you would do then would be do a second run where you take whatever you're injecting in and then just inject it into the buffer. And then you can actually directly measure what the heat of dilution is. And also, you need to kind of think, well, did I actually match the buffers? In this case here, it was actually DMSO. And as I kind of mentioned in earlier talks, having accurate concentrations of DMSO can be quite tricky. So here's a very, very nice example. In red, there you can see you're injecting the protein, you get a nice profile. And then the second, you do it into this peptide here, and put this in the buffer. And so you get 2 heats here, and you essentially just take that baseline away and fit it, as we'll see in a second. Okay, so what it does is then all those peaks, it integrates the power difference in all these injections. It normalizes then relative to the moles of ligands injected. It adjusts for the heat of dilution of the ligands. So you imagine you've got a fixed volume of cell, and then you're injecting small volumes in. So then the volume at the end is going to be bigger than the volume you actually started with. So that's it needs to adjust for that. Normally now we just fit in peak software, but previously then we'd fit in Origin. You can export things in nitpick and fit in SEDFit. So typically then we get raw data, we correct it for the baseline, and then we get this kind of characteristic trace here, which is this kind of sigmoidal curve. And at the bottom then, the x-axis is molar ratio. That's the molar ratio of the injectant over what's in the cell. And in very, very nice graphs, then what you can get is from the bottom of the S there, you can get then what the delta H is up to the top. And then in the middle, then you'll get a stoichiometry. And then what that slope looks like, then will tell you what the equilibrium constant is. And of course, by doing non-linearity squares, you get stoichiometry, you can get KD and the delta H. And then we can use those then to get what the delta H is, so the delta G is, and then we can then calculate what the entropy change is. Okay, well, what happens if you get a stoichiometry of 0.8? What does that mean? I mean, is 0.8 of my protein binding onto 0.8 of the other protein? It doesn't make much sense. One thing could be, so your concentrations aren't quite accurate. Another thing could be that actually what you're doing is you're showing how much active material you've got. So maybe 20% of your material isn't actually active. So we could actually just view this is actually a number, the number of active sites. So if say you get a value of 0.5, that has a real meaning. 0.8, we just assume that that's a one-to-one interaction. And of course, if you get something that's above 1, like 1.7 or 2, then that tells you there might be 2 binding sites. Do you want 2 minutes history? Anybody have 2 minutes history? Okay. These are the actual founders of this field, calorimeters, and you can see there that this is kind of bulk coloring of the pads, these are the X-rays, and so it's a lovely picture by David. So, it's also a very interesting picture as well, because actually so Marianne Polez is standing up, she's looking at it as well, and her husband's kind of sitting down and actually writing, looking at her. So they were the founders. Just before the the French Revolution. He unfortunately was a tax collector, so it's come the revolution. And so Marie Anne is very, very important because not only did she translate all the text into French, she was also she worked in the laboratory, and she also was the founder of really the scientific method in the fact that she actually drew all the equipment in for their journals so that then everybody else could see what they developed. And during the French Revolution, she didn't get guillotined, and she actually saved all the work. Right. So let's get back to the science. So let's think. We didn't get a really good kind of results. We didn't get see this S curve. So what can we do? Well, two things. We could change the concentrations and the temperature. Okay, so the concentrations. So then on the left there, that's kind of a nice typical graph. And we say it's got a C value of 7. And what do we mean by that? Well, we mean that that's the concentration in the cell above the KD. Because we're doing stoichiometric titrations, we actually want a higher concentration in the cell above the KD. So it's kind of the optimal range is between 5 and 50. And then the optimal range for this range is going to be 10 to 15-fold above that. Depending upon what the C value is, how much the concentration is above the KD, you get these different kind of curves. And we'll kind of look at these just in a second. So if we have a concentration in the cell way above 500, in purple there you can see it's 1,000. That means you're going to be way above the KD. You get these very, very sharp transitions. So in purple you can see what's happening is ligands going in, binding, and then suddenly you get a step up, everything's basically bound. So you kind of think, well, why can't you just lower the concentration? Lower the concentration. If it's say like 10 nanomolar KD, well, why can't we just use 100 nanomolar? The problem is then you get less protein in the cells, you get less number of sites, then the less number of reactions and the less heat. So it's kind of a lower limit, about 5 micromolar to kind of measure this kind of total heat. So to get around this, then what you can do is you can then, if you've got a weaker binding ligand, which you know what the KD is, you can form a complex with that and then displace it with the tighter binding ligand, and then just do the calculations to get what the tighter binding ligand is. If you've got very weak binding, so say your C is less than 10, say C in yellow, it's C is 1. Well, what do you get? You don't get this kind of nice S curve. You just get this gradual thing going up. And that means that it becomes difficult to find, well, what's the KD? What's the end point? You can get delta H. Then in these kind of cases, it's really important that you make sure that you've got your ligand buffer control. And to get KD here, you could then do a displacement where you're going to displace the relatively, again, form this complex with weak interaction, and then try and displace it with a tighter KD where you know. So what about temperature? Well, as kind of mentioned earlier on, delta Cp means delta H kind of changes with temperature. And so here on the right, you can see as we increase the temperature, the delta H becomes much, much more negative. And then on the left, you can see start off at 12, we increase the temperature, and you get much bigger delta H. Of course, if you think about this as well, if you say you have an thermic reaction where heat's given off, then if you cool it down, you can get much bigger positive delta H. Okay, additional uses you can kind of do. Well, you can measure oligomerization. So you have your protein is an oligomer at high concentration, injected into buffer, and you can actually follow that process as well. And then we'll look at some other just examples of like protonation changes, steady state kinetics, and actually pre-steady state kinetics. Okay, this is another thing that happens during reactions. Imagine you're forming an interface. And of course, residues within that interface, the pKa may actually change upon forming that. So what happens then if you're at 1 pH and then you get a change in pKa of the protein? Well, you might then get a protein released. Well, protein released, that's going to change the pH. The buffer is going to oppose that. So there you're going to have on the top there, you're going to have another reaction. And so that then the actual observed heat down at the bottom, it's not going to only contain the interaction heat, but also the ionization heat. But not all buffers are the same. So here's a table here, and as you can see, lots of different buffers. And you can see they have in the delta H ionization here in the middle, and you see the vast difference between all of them. So if we want to kind of get into kind of seeing the difference of protonation in proteins, well, we can actually choose 3 different buffers with different ionizations and then compare the reaction here. Point to note as well, if you look at the right-hand column here, it's the delta H and delta T. You can see there's actually a big change for some. If you look at tris, there you can see there's actually quite a big dependence on temperature and what this ionization energy is. So that when you change the temperature, the actual pH of the buffer changes as well. So tris is notorious for that, of not keeping the same temperature. You make it at room temperature, take it into the cold room, it's a different pH. So always be careful about that. Okay, so let's just choose these 3 buffers and then do this reaction again. And they all have a similar kind of pKa of buffers. So this is then a reaction here, and then you can see in green it's with tris, and then in MOPS in purple, and phosphate in orange. Yeah, lots of different then heats coming off. And so if we then measure the observed delta H, and then track that against what the ionization is, then we can actually find out what the protonation is during this reaction at all. Of course, if there wasn't any protonation happening, then this line would just be flat. So additional uses as well would be using enzyme kinetics to steady state. First one you would do would just be high enzyme concentration and very low substrate. So you just trying to get the heat of the turnover. And then they would do a kind of a normal kind of enzyme reaction. You get a very, very low concentration of enzyme, and then a high heat, a high substrate concentration. And then you're looking at the heat generated at each of these substrate, and that's going to be proportional to the rate of the reaction. And so if you plot that against one over the enthalpy, you can then get a McLaughlin. And another more recent use of ITC is people kind of noticed when you inject in, the actual shape of the peaks at the start and at the finish are quite a bit different. And what's happening there is that the number of sites that are actually free are becoming less and less. So if we imagine that the on and the off rates, or the observed on rate here, that's going to be dependent on the K on of the 3, say, A and B kind of going together. So if there's less, say, sites available on, say, A, then that observed rate is going to slow down. So the actual time to get back to baseline is going to lengthen. And they find that if you then plot this, using this equation here, don't worry about that, you'll get a curve here, which you can then fit to give you what the K off is. And of course, if you know the K off, you know what the binding constant is, then you can work out what the on rate is. So here's some examples of ITC that we've done. This was a few years ago then. This is a crystal structure from Mark van Bruegel. Excellent crystal structure, and the reviewers went, yeah, but is it real? You know, it's this kind of coil. Yeah, do they just form at kind of high concentrations anyway? You know, is this, prove to us that it's actually real? So then what we did then is they made mutations, and you can see across the interface here, in both the coil in the center then, and the globular domains. And top here, you can see in wild type, you're getting a nice heats, and gray is buffer control. And then when you make mutations on one protein, that just destroys the interaction completely. And then the bottom here, when you make mutations on the other side of the interface, And then you destroy the interaction as well. So then that showed that was proof. And now here's something that's a bit unusual, which Chris did very, very recently. And this was working with Tom May's group. And they work on these, they're kind of, are they archaebacteria? And this lives in the sea. I mean, there are things at the top of Tunis. And what they find is that they use ammonia in their metabolism. And they're very tough beasts because they've got this S layer going around them. And so what is that they then take the ammonia in and concentrate it near the membrane for metabolism. And what you can see then in black is that when you put these bacteria in the cell and you inject in ammonia, you actually get some heat coming off. If you start to destroy this S layer, then by putting EGTA, you can see the heat go down. And on the center there, you can see then the heats are being destroyed. So calorimeters, they can measure anything. Okay, so in the last little section, we're going to then go from isothermal then to scanning. So with scanning, then what we're doing is then we're taking the system and then we're increasing the heat such that we will actually denature. And again, if you remember back then, okay, you're looking at this biological process, but isn't there kind of high delta CPs, changes in the heat capacity, and will complicate things? And that does complicate things quite a lot. So again, don't worry about the equation itself. Just realize that actually you're going to get curvature in the thermal stability here. And so what you can see here this is temperature down at the bottom, and this is stability. So anything above the zero, then that's stable. Anything below that's negative, then that's unstable. And this curvature then leads to two things. Okay, there's a maximum stability where the delta S is not. And as you increase the temperature, then you'll get down to this point here where the delta G is 0, and then half the material will be unfolded. So this is called the TM. But there is a consequence, in fact, of this curvature. If we then cool things all the way down, then actually at the bottom there, you can see in green particularly, you're actually going to get this unfolding at cold temperatures. So thankfully, most proteins, then this temperature where it cools and folds is way below 0. But for some things, your protein could be not very stable at cold temperatures. So always be careful. And here and then in the different colors is then the different changes in delta CP. And you can see how that changes then where this it had all the same TM. You see how this changes where the coldest melting is. So what does it look like? Well, it looks pretty similar to an ITC, except for there's no injector. You've got two cells, one with a reference, one with a sample. And again, it's trying to kind of keep the temperature difference the same between the two cells. But this time you're going to have a main heater, which is going to ramp up the temperature of both those cells as well. So it's again got feedback to try and keep this temperature the same. You know, we've got a robotic one, so it's just automatic loading. The volume, you need about 130 in the active cell, but to actually load it properly, then you need 360 microliters of this kind of concentration. The temperature can get up to 125. That's beyond boiling. So therefore, we've applied a bit of pressure on there to get that temperature up. Variable scan rates. And what's going to give you is going to give thermal stability, the TM, and also the enthalpy. So you can actually then look at mutants and various conditions. So this might be kind of a typical kind of trace. So you can see it's measuring then the delta, the CP, so that's the heat capacity. How much heat do we need, does the system need to put in to actually raise the temperature up? So it's measuring a direct measurement of that. And so you can see then there's a kind of a baseline and kind of is going up. And then you get unfolding, and then you get a spike where the TM is, and then it comes down again, and then all the other materials then unfolded. And then if you can compare, you can see that actually between the native and the unfolded, there's actually a slight difference in what the heat capacity is. And then that's something I often don't know if that's going to be positive, but it's all a bit slopey. And that's why we also need to do buffer baselines as well to actually be able to correct. So here's a case here at the top where you've got it's slightly assigned with different pHs, and you can see again, now you can actually definitely see the difference in the heat capacity before and after unfolding. And there in the inset then we can see the pH dependence of the melting temperature. So what do we use it for? Well, we can do photostability here, or we can do ligand binding. Down at the bottom there, then there is small nucleotide, then binding onto a protein. And as you can see, as you add more and more of that in, increasing stability. So it's not only kind of used for just kind of simple kind of undilate proteins, but it's used a lot in, say, formulation when you have like antibodies that need to be at very, very high concentrations. And it can use in very, very complex samples, which I'll show you just in a second. Okay, so here at the top here is also on the left. Then we've got a very complex sample. We've got plasma. So at the bottom, you can see this would be human plasma proteins, and between 40 and about 90. It's a very, very complex kind of profile. And so what this group has actually shown that if you then do the individual plasma proteins individually, and that's in the top trace there, you can actually see that if you just add these all together, you end up with a trace at the minute. And what they find for individuals, and for well, actually these traces are pretty much the same if you average over them. But what was very interesting, and I think this is what they're developing, is that in diseases on the right-hand side here, you get different profiles, and actually different diseases have got different characteristic traces here. And this is not because there's different levels of protein. What they think is actually going on is in these different diseases, then there may be either difference in metabolism or different in, say, release factors or things that are breaking down. And that's they may all be sticking to all the plasma proteins, and then that's kind of changing these kind of traces. So I'm going to end it there today and tell you that, okay, more information and more detailed information is then in our book, Protein Ligand Interactions. Chris Johnson's chapter on ITC. He also has done a review recently in Nature Methods Primers, and also he has an article on DFC, then in the Archives of Biophysics and Biochemistry and Biophysics. And of course, just come and talk to me and Chris if you kind of are interested in doing any of these techniques. So thanks for your attention today, and any questions?